So day 2, you're going to make your drug treatment groups. So I'm using the 10 millimolar stock solution of AJA, azelaic acid, which is 10,000 micromolars. And I need 20, 40, and 60 micromolar concentration for my drug treatments. Each treatment has 3 wells, and I'm going to put 2.5 milliliters per well, which is 7.5 milliliters of each treatment. But remember, we always make extra, so I'm going to do 8 milliliters. So to find how much of the drug I need to put in, I'm going to do my 20 micromolar, which is the concentration that I want, and 8,000 microliters, which is the volume I want. And then on the other side I have the concentration of the stock solution. We're going to find how many microliters of the stock solution we need. So that gives me 16, and then do the same thing for the 40 and the 60. So I need to add 16 and 32 and 48. Okay, so I'm going to add that. The wrong way. That was open the wrong way. Okay, well, we'll just start over. Since that first one was the wrong way, I couldn't use it. I had to dispose of it. There we go, because you can't touch the tip. So I'm going to put 8 milliliters of media in each of my tubes. My tubes are already labeled. That was the untreated. Okay, so for the next 2, I'm going to pull the volume for both of them up in here. So I'm going to take 16 and then just let out 8 at a time. So try and do that whenever possible so we can save pipettes. And just, the trick is just to let it out really slow by not holding the button down quite as hard. And then you'll be able to read the numbers just fine. There we go. Okay, so now the untreated one is complete because there's not going to be any drug in it. The 20 micromolar, remember I said I'm adding 16 microliters of drug. So the first thing you need to do is take out 16 microliters because we still want our total volume to be 8 milliliters. So I'm just going to eject the tip with the media into my beaker. So then I will make sure my drug's all mixed up. Take out my 16 microliters. And when you put it into the tube, make sure you're in the media and go up and down a couple times to make sure all the drug has come loose. So that one's complete, and I like to move them back once they're complete and tighten the lid so I don't get them confused. The next one was 32 microliters. Again, I'm going to take out 32 microliters and get rid of the tip, and then put in 32 microliters of my drug. Always check and make sure there aren't any bubbles. Sometimes if you pull up on the stopper too quickly, you'll get bubbles in there and you won't have an accurate volume. And the last one is 48. So I just took my plate out of the incubator and I already looked at each individual well under the microscope and make notes about how confluent the cells are and if there's any noticeable differences. So I'm going to do one treatment group at a time, which is 3 wells. You don't want to leave any of the wells without media for too long. So I'm going to do, just slide to the pipette down the back of the well and just gently touch the bottom of the well. Because remember, your cells are stuck on the bottom, and so you do not want to vacuum them off. Alright, you can use the same vacuum pipette as long as they're all the same cell line. And I haven't, yeah, make sure it doesn't touch anything, so you can just set it right back in there. Okay, so these first are my untreated. I'm going to loosen the lid, and I can pull up, using one pipette, I can pull up 7.5 and then let out 2.5 at a time. Make sure you turn it where you can easily see the numbers. Okay, so this part is important. When you're letting the media out, make sure to gently touch the edge of the well and then slowly release the liquid. This makes sure that you're not knocking off the cells in the bottom of the well. Okay, and then move on to the next group. Oh, sorry, I already have one. For all your treatment groups, remember your drug is in there, so make sure and invert and make sure the drug is equally mixed before you put it in. Alright, so put the cover back on, and it's always good to look at your cells one more time under the microscope and then put them in the incubator overnight.